Developing reliable molecular biomarkers for mesothelioma poses significant challenges. Mesothelioma is an aggressive cancer directly linked to asbestos exposure. Asbestos was widely used in construction and insulation materials for much of the 20th century before its carcinogenic properties were fully known. Even decades after exposure, asbestos fibers can remain in the lungs and pleura, continuing to cause DNA damage and malignant transformations. The long latency period between initial exposure and cancer development complicates identifying definitive diagnostic and prognostic biomarkers. Mesothelioma tumors can take 30 to 50 years to form after asbestos inhalation. During this extended incubation time, numerous genetic and epigenetic changes accumulate that drive the malignant transformation. Pinpointing the specific molecular alterations involved in initiation and progression is challenging given the multitude of insults from asbestos fibers and normal aging processes over many decades. Mesothelioma arises from the mesothelial cells lining the pleura, peritoneum and pericardium. Obtaining high-quality fresh frozen tumor samples from these sites for comprehensive molecular profiling has proven difficult. The tumors are often diagnosed at later stages when they have spread. Surgeries to obtain intact tumor tissues risk disturbing malignant cells and promoting further metastasis. As a result, most tissue samples available for research have undergone some ischemic damage or contain mixed populations of normal and cancerous cells. This complicates efforts to detect subtle changes unique to the malignant cells. Three main histological subtypes of mesothelioma exist: epithelioid, sarcomatoid and biphasic, with significant biological heterogeneity both within and between these classifications. Epithelioid mesothelioma generally has a better prognosis than sarcomatoid or biphasic variants. But even within epithelioid mesothelioma, survival durations vary widely. Pinpointing precise molecular determinants of clinical behavior in each subtype has been challenging due to limitations in sample sizes for comprehensive profiling in large clinical cohorts. Reliable biomarkers would need to detect subtype-specific alterations as well as those common across all mesotheliomas. The rarity of the disease also poses challenges for biomarker discovery. Despite asbestos bans in many nations, yearly mesothelioma incidence rates remain relatively stable or are increasing as past exposures manifest clinically. Nonetheless, mesothelioma accounts for only a small percentage of cancer cases worldwide, around 3,000 per year in the United States alone. This limits opportunities for researchers to obtain sufficient sample sizes to robustly identify molecular signatures that differentiate malignant from non-malignant states in large patient populations. Statistical power is reduced for detection of subtle but clinically meaningful markers. Multi-institutional collaborations are needed, but differences in sample collection, handling and clinical data across sites can complicate harmonized analysis. A further difficulty is that mesothelioma is etiologically distinct from other lung cancers. The vast majority of mesotheliomas are directly linked to asbestos exposure rather than generic lung carcinogens like tobacco smoke. This means generic lung cancer biomarkers may have limited relevance for mesothelioma. Dedicated studies are needed focusing specifically on asbestos-induced disease mechanisms. Funding opportunities more often support research on more common cancers rather than rare diseases like mesothelioma. Sufficient resources for large-scale, appropriately powered biomarker discovery trials are lacking. The multi-step nature of malignant transformation also challenges biomarker discovery. Mesothelioma development likely involves successive cooperation between diverse genetic and epigenetic alterations, making it difficult to pinpoint any single defining molecular change. Early diagnostic biomarkers may need to detect subtle alterations occurring far back in the disease evolution, before clinically overt cancers emerge several decades later. A combination or panel of molecular alterations assessed together may prove more reliable than any single marker, but developing robust multiparametric assays adds further complexity. Longitudinal studies tracking high-risk asbestos-exposed individuals from early exposures through disease progression could shed light on the temporal sequence of molecular changes driving mesothelioma. Such studies require immense resources, long study durations and sample preservation to achieve sufficient statistical power when dealing with diseases with long and variable latency periods like mesothelioma. Overall, the complexity of mesothelioma pathogenesis from initial asbestos insult to clinical cancer, limitations in sample availability and disease rarity present major obstacles to establishing reliable molecular biomarkers, though continued collaborative research efforts seek to overcome these challenges. Developing robust and clinically useful molecular biomarkers for mesothelioma diagnosis, prognosis and therapy monitoring faces substantial difficulties related to the long disease latency, heterogeneity within subtypes, difficulty obtaining high-quality samples, rarity of the disease and our incomplete understanding of its precise molecular pathogenesis. Larger collaborative studies and dedicated research focus and funding are still needed to address these challenges and realize the potential of molecular diagnostics to improve mesothelioma patient care and outcomes.